Hey, Mr. P here. In this video, we're going to talk about transport in the xylem of plants. So the xylem are the vessels or part of the vascular tissue that water is going to flow through. And we've learned a little bit about vascular and non-vascular plants, but now we're going to talk about how vascular plants specifically transport water through a kind of continuous stream of water from roots to shoots in a process called transpiration. And in order for us to understand what transpiration is, we need to first understand how gas is exchanged in a leaf. The leaf is a part of the plant that undergoes all of the photosynthetic activity. That's where all of the gas exchange happens. And when we say gas exchange, we're talking about carbon dioxide and oxygen. Oxygen is a gas that is a byproduct of photosynthesis, and CO2 or carbon dioxide is a reactant of photosynthesis. And so there are these little structures called stomata that we'll talk about in a minute, but these are the openings that allow these gases to exchange. And when I say exchange, I mean carbon dioxide being a reactant must go in, and O2 or oxygen being a byproduct or a product of photosynthesis has to be removed. So what are these layers that are indicative of leaf tissue? First, on the very outside, you have a cuticle, and the cuticle, sometimes called a waxy cuticle, is a waxy layer that protects the plant against water loss, being the transpiration stream, and also helps protect the plant, the really important plant tissue, from UV radiation. This is kind of the sunscreen layer, so to speak. And so it's a waxy cuticle layer that protects the plant against water loss and insect invasion. It makes the plants less palatable from insect invasion, but it also helps protect against the UV exposure, which we know can actually cause DNA mutations. We obviously don't want plant tissue to undergo DNA mutations, which can result from the UV exposure. Next, you have a layer called the palisade mesophyll. This is the upper mesophyll. Mesophyll being kind of this entire interior portion of the leaf. But the palisade mesophyll is going to contain large numbers of chloroplasts, which are those organelles that are responsible for photosynthesis. And so if you have a large number of these chloroplasts, you're going to be able to carry out photosynthesis in a very efficient manner. This layer is towards the top of the leaf where light is most available. Obviously, the light is going to be coming in, penetrating a little bit into the leaf tissue, but as long as it penetrates enough to reach this palisade mesophyll layer, where all of these little chloroplasts containing chlorophyll are located, that's going to be necessary in order to photosynthesize. The light is not going to penetrate as much deeper into the spongy mesophyll, which is why the plant wants to kind of congregate all of its chloroplasts towards the top, where sunlight and visible light can penetrate. But the spongy mesophyll down here does have a role, and it does have a significant role, in the utilization of the gases for photosynthesis. And so it consists of loosely packed cells with very few chloroplasts. Its job is not to actually absorb the light like the palisade mesophyll, but it is designed to have many air spaces in which to provide an increased surface area for gas exchange. You'll notice that this layer is much more loosely packed. This one is much more concentrated with cells. This is where the light and the actual active photosynthesis will take place. This is where light is absorbed. This is where the gases are used. But the spongy mesophyll is all about gas exchange. Okay, it's getting CO2 to this layer, and it's getting oxygen out of that layer. It needs to be loosely packed because these cells need to be able to touch air, right? Air comes in through these stomata, these openings, and it has to contact these cells. It opens up a lot of surface area, decreases a lot of volume, and it allows for gas exchange to take place. A vein, which you can see in this particular cross-section, is going to be vascular tissue, just like veins are in us, okay, or in other organisms. But the veins in plants are vascular tissue. They are always located near the middle of the leaf to be closely associated with all tissue layers. The xylem and phloem both make up portions of the vein. The xylem are going to be the vessels that specifically transport water. The phloem are vessels that specifically transport sap, which contains the sugar, which are the photosynthetic byproducts. Next, we have the stomata. We talked about them briefly. Those are the actual physical openings. We call them stomatopores or stomata, but they occur on the bottom surface of the leaves that allows oxygen to diffuse out. It allows carbon dioxide to diffuse in. Basically, the exchange of those two gases in the leaf and the surrounding environment. They're also associated with the bottom of the leaf where light intensity is the lowest. Remember, light is striking the leaf from the top, not striking the bottom. And so it correlates to a lower temperature because it is not heating up due to light exposure, and it is also not exposed to as high or as intense of UV radiation. Okay, this is kind of the protected layer or protected side of the leaf. This is the exposed side of the leaf. Okay, all those work in a process called gas exchange, and as that process of gas exchange occurs, it is an inevitable consequence that water is lost from the plant because water is going to be lost alongside the gases that are exchanging. Because remember, water is in a vapor, it's in a gas form as well, and so as long as there is an opening from the inside of a plant to outside of the plant to exchange these gases, water is going to be a gas that is lost as well. As a result of this water loss, plants have developed specialized cells called guard cells, which we'll talk about next, to control the opening and closing the stomata to ultimately control water loss. Okay, so what are guard cells? Guard cells are these actual physical cells that are guarding the opening of a plant. And if you look at them, obviously in an artistic rendition, they look more like this, but when you look at them actually through a microscope, they will look like this, very similar. They can be open like you see in this particular image, but they can close in times of water scarcity as well. So what are the guard cells really, and how do they work? Stomata can only be closed on a short-term basis, which means they are by default open. Okay? Stomata are always open by default. They can close, however, on short-term basis because the plant needs to conserve water. This is the only time they would close. But they're open by default because the plant needs a constant supply of carbon dioxide so that photosynthesis occur. Photosynthesis, as we've talked about in past topics, is 6 CO2 plus 6 H2O yields in the process of light and chlorophyll C6H12O6 plus 6 O2. That is the photosynthetic equation, and you can see that CO2 is a reactant, water is a reactant. Those two things need to come into the plant so that the plant can obviously make glucose, but also will release some oxygen as a byproduct as well. In order to constantly supply the cells with CO2, these stomata have to be open so that carbon dioxide can enter. Stomata open and close because of turgor pressure. The turgor pressure is the amount of pressure inside the plant cell based on it being hydrated. Okay, if there is enough water inside this plant cell and the water is in enough concentration that it's constantly increasing the pressure on the inside, the pressure is going to constantly be pushing on all walls of the cell, and it's going to create a rigid cell wall. When plants become less turgor or turgid, they're going to start wilting, which is why plants start drooping. They start getting a little more flexible. That's obviously what happens in response to low water concentrations. Guard cells have unequal cell wall thickness. You can actually see that in this particular image. They're going to have thicker cell walls on the insides of these stomata and thinner walls on the outside. Okay, that is an actual trait that is really important to the proper functioning of these cells. When cells take in water, they bulge more. As I said here, when they take in water, they're going to increase the pressure and they're going to bulge more from inside out. When they do that, they're going to open the stomata, and it's going to cause the opening to increase because the water is basically pushing on the thinner side, causing this to flex out because these thicker cell walls are going to be a lot more rigid and a lot stronger than the outside walls. So the outside walls give, not the inside walls, and it pushes the entire thing out, and it allows the stoma to open as a result of increased water concentration. You can see that these vacuoles are a lot bigger because there's a lot more water in these, which correlates to the stomata being open, whereas in this picture, these vacuoles are much smaller. There's much fewer water molecules, and so because there isn't as much turgor pressure, they're not pushing on these walls, and the stomata closes. Okay, when cells lose water, they sag towards each other, closing the stomata. Another thing that you want to kind of think about is these stomata are going to remain closed unless there's enough water to open them. That's how I like to remember it. If you think about the stomata being kind of just closed when water is scarce, it makes sense because they don't want to lose water. Okay, so it wants to conserve water, it closes. As water starts coming in, it starts increasing the pressure on the inside, and as the pressure increases, it's going to force these out because they're swelling with water, and it opens the stomata. Okay? So, how do these guard cells work other than just increasing and decreasing in water concentrations? They actually have to do with these potassium ions. And if you look in this back picture, you can see that there are potassium ions in these particular diagrams as well. You'll see that in times of water availability, more of the potassium ions are on the inside. When water is scarce, a lot more of the potassium ions are on the outside. So, what does that mean? The gain and loss of water in the guard cells is largely because of the transport of potassium ions. When water is scarce, potassium ions are out. When water is available, potassium ions are in. The blue part of the light spectrum, remember that visible light spectrum is Roy G. Biv, and so it contains a blue frequency as well as red frequencies, but the blue specific frequencies, the blue part of the light spectrum, triggers the activity of an ATP-powered proton pump in the membrane of the guard cells, which causes the active transport of potassium ions into the cell. So when the plant is in the presence of blue light, if I change this, when the guard cells are in the presence of blue light, and blue light is coming in and striking these stomata, it's going to cause a proton pump to actively transport the potassium ions into the cell. You'll see these potassium ions now coming into the cell as the blue light turns on those proton pumps, and it's going to cause potassium ions to come in. Because the potassium ions are now in, we have a higher solute concentration inside, causing an inward net movement of water by osmosis. If we make inside more hypertonic, water always wants to go hypo to hyper, and so water is going to come into the cell. It's osmosis, okay? Increase the solutes inside, move the water as a result of that. When potassium ions passively leave the cells, water also leaves. So in times of no blue light, if there isn't blue light, and I kill this light, right? No more blue light, then all of a sudden there are no proton pumps. These potassium ions have to be basically shuttled out, and because they're shuttling out, this becomes hypertonic side, and therefore the water leaves as a result of all of those solute molecules moving. The water follows the solute, right? Water is going to follow the solute, and in this case, the solute is the potassium ions. When potassium ions passively leave the cell, water also leaves, and there is a plant hormone called abscisic acid, which can cause potassium ions to diffuse rapidly out of the guard cells. Abscisic acid is specifically produced in the roots during times of water deficiency, and so it makes sense that if the roots are producing this growth hormone called abscisic acid, when the roots know the soil is dry, it's going to cause a really fast movement of potassium ions out of the guard cells, which is going to ultimately pull the water with it. All right, so now we've talked a little bit about how the guard cells work, a little bit about the cross-section of a leaf, talking about gas exchange, and now we're going to focus on the vessels that I talked about in that cross-section. The vessels, or the vascular tissue, is made up of xylem vessels and phloem vessels. The purpose of this video is talking about xylem vessels specifically, and so we're going to talk about how the plant moves water through its tissue. Water molecules have cohesive properties due to the hydrogen bonding and polarity. We've talked about those when we talked about water early on in our IB syllabus. The way these water molecules work is that there is a hydrogen molecule side. Okay, both of the hydrogens are always on one side of the oxygen. They give a slightly positive net charge, and then you have a slightly negative net charge on the oxygen side. And the attraction between the slightly negative and slightly positive side of water molecules is called hydrogen bonds. It's a weak bond, but it forms a hydrogen bonds, and it causes water molecules to stick together. We call this cohesion, but these hydrogen bonds can form between these water molecules and other surfaces as well. We call that adhesion. Okay? You have to remember that. There are cohesive forces and there are adhesive forces. Both of them are the result of water being polar, and they're also the result of hydrogen bonding. Hydrogen bonding also due to water being polar. Plants transport water from the roots to the leaves to replace the losses from transpiration. Water, as you probably are aware, goes from the ground into the roots, through the root system, through the xylem vessels, through the plant, through the stems and twigs and branches, and through the leaves, and is ultimately lost through the leaves, through those stomatophores, in a one-way flow of water from roots to shoots. And so water potential gradient is going to be decreasing as we go higher above the ground. But there is a one-way flow of water from the ground to the atmosphere through the plant. That is called transpiration. Okay? So cohesion and adhesion in xylem, you see them both. We just talked about water molecules forming hydrogen bonds with other water molecules. We call that cohesion. We also talked about water molecules forming the hydrogen bonds between water molecules and the vessel wall in this case. That is adhesion. Both of them are the result of polarity, and they're both the result of hydrogen bonding. And so because we have water molecules attached to all water molecules in this chain, it causes a chain of water molecules that are constantly being flown up from the ground to the environment, and it creates a certain tension that is created by this transpiration stream or this transpiration pull. Tension occurs in the columns of water in the xylem vessels because the water is constantly being removed by evaporation. There is water molecule that is constantly being evaporated out of the plant, and as one water molecule is evaporated, it pulls the entire column of water up with it, which in turn pulls a water molecule into the roots from the soil. And so we constantly replace water that is lost through evaporation. That is called transpiration. It is really important in fully understanding not only how plants remain hydrated, but also how plants constantly replace the water they lose through the photosynthetic pathway. The water columns remain continuous because of the cohesion and adhesion. Water molecules are constantly attached to other water molecules that creates a continuous column of water. At the root level, you have root structure that allows for water to be brought in. You have the soil around the roots, you have these root hairs, and water molecules are constantly being absorbed by these roots because of, again, this transpiration pull. As transpiration is pulling this column of water up towards the shoots, it is creating a tension that is sucking the water into the roots from the environment in order to keep that stream of water continuous. Roots function to provide mineral, ion, and water uptake for the plant. This is how the plants get in their nutrients and their minerals and their water. Water moves into the root hairs from the soil because the root hairs have a higher solute concentration than they do water. And so because they have a lower water concentration than the surrounding soil, the water is going to diffuse into the cells. It's called osmosis. We see this several times throughout the plant structure. Osmosis is kind of a fundamental process of getting water into a plant. And there are some zones that we need to talk about when we talk about root structure. This zone of cell division right here towards the root cap is going to be new undifferentiated cells. This is active cell division. That's how the root is growing. Then you have a zone of elongation. Cells are enlarging in size corresponding with the G1 phase of the cell cycle. They are not dividing in this stage. They would have already divided, but they're growing, getting bigger, and so they are elongating in this stage. And the zone of maturation is upstream a little bit from the root cap. Cells become a functional part of the plant. They have reached adult size. They're not dividing anymore. They're just maintaining their normal processing at that stage. Each of the root hairs is a little small projection that comes off of the main root or the main tap root sideways. They are designed to increase surface area, decrease volume. They aid in water and mineral ion absorption. It's how we get water and minerals into our plant. And then you have a root cap. The root cap is the protective covering that is on or protecting the apical meristem, which we'll talk about in more depth later. During primary growth of the root through the soil, because this root is growing down through the soil, which may be abrasive, it may be rocky, it may be clay, right? It has to protect this really, really important apical meristem tissue, which is the undifferentiated kind of bunch of stem cells. It has to have a cap so that as it pushes through the soil or pushes through the substrate, it is not damaging the apical meristem. Okay? New undifferentiated cells are forming in this particular apical meristem. That is a really important part of the plant. Mineral ion movement. It is essential that mineral ions move into the root as well as water. We obviously don't want just water to move into the roots. We want mineral ions. That's how the plant kind of gains the building blocks of life that it needs. Proton pump generates differences in the H+ ion concentration and electric charge across the membrane. These proton pumps are a form of active transport. You'll see that these H+ ions are going from low concentration to high concentration, from inside to out. That is an active transport. It requires ATP. ATP is going to come in, power those proton pumps so that H+ ions can move. The difference in electric charge causes the cations, such as the potassium ions, to enter the cell. Okay, so this is a potassium channel. They are going from low to high, from outside to inside. That is also a form of active transport, but it is using the flow of H+ ions to power the potassium channel. It's not using ATP in this case. And then you have transport proteins that are kind of co-transporters that are transporting, in this case, chloride ions into the cell with the movement of these H+ ions. So you're still moving them from low to high, but we're moving them with the aid of the H+ ions, or with the passive transport of the H+ ions into the cell. Okay? The xylem are vessels. We just talked about them, okay? But a little bit more about the actual anatomy of the vessels. The xylem are the vessels that transport water, okay? They are going to be oriented in bundles called vascular bundles. Vascular bundles not only composed of xylem, they are also composed of phloem. Xylem cells contain no cytoplasm, and so they are therefore non-living remnants of cells. They're basically non-living vessels that have no cytoplasm, which makes water transport more efficient. They're basically just hollow straws that have kind of remnants of the cell wall, which created the vessel to begin with, but they are not alive. Xylem cells move water via a passive process called transpiration. Cell walls are thickened and incorporate lignin, which are spirals or rings, which is a protein that helps fortify those cell walls to make them stronger and in order to withstand very low internal pressures without collapsing. There is a lower pressure on the inside of these vessels than there is in the atmosphere around them. And so because there is lower pressure, the pressure on the outside wants to force in and collapse those cell walls. So we have to fortify them with these spiral proteins, or these rings of protein that kind of go around the outside walls, which fortifies them. That protein is called lignin. Xylem cells are arranged end to end to form a continuous vessel. The walls between cells have been reduced, which makes water transport easier, and they have these little kind of fortified conjunction points, but they are open from one cell to another. Again, they're not living, but it allows a continuous flow of water from the root to the shoot all the time. It's never broken. Okay. So what about plant adaptations for water conservation? Because some plants live in environments that are really, really water deficient. Some plants live in water, and they have a constant supply of water, but some plants, like your cacti or your succulents, don't really have much water to work with at all, so they have to conserve water. There are a variety of environmental factors that I think you should know, one of them being light. The effect of light is going to speed up transpiration by warming the leaf and opening the stomata. So if you add a very intense light to a plant, it's going to speed up transpiration because it's going to speed up the temperature, or it's going to increase temp, and it's therefore going to speed up transpiration loss. Humidity. Humidity is actually going to decrease transpiration. So if we decrease humidity, it's going to increase transpiration. They are inversely related. If you increase humidity, or if you have plants in a really humid environment, it's going to decrease transpiration loss because there isn't as big of difference between the outside water concentration and the inside water concentration. If you drop humidity and decrease humidity a lot, it's going to therefore increase the speed of transpiration loss, okay? Because there's a greater difference in the water concentration both inside and outside of the plant. Wind is going to increase the rate of transpiration because it's going to pull the humid air next to the stomata away, decreasing, in effect, the humidity next to the plant, and that is going to increase the transpiration pull. Temperature is going to increase if the temperature increases the transpiration loss, but it is also going to decrease transpiration loss if temperature drops. Those are basically directly related. Soil water, if you increase the amount of water available to the plant, it's going to increase the intake of water, and so it's therefore going to cause the plant to become more turgid, which is going to cause the stomata to open, which is going to increase the transpiration rate. Carbon dioxide is a reactant for photosynthesis. So high carbon dioxide levels in the air around the plant usually cause the guard cells to lose turgor and stomata to close. So high carbon dioxide concentrations can actually cause transpiration to be reduced. All right. Last thing we need to talk about are the xerophytes. Okay, xerophytes are awesome. They have a ton of adaptations to conserve water loss. These are the cacti and the succulents that I talked about in the last slide. So what are some of the adaptations that they have identified or that they have created in order to minimize water loss? They have small, thick leaves reduced the surface area and thus decrease water loss. These spines on a cactus are actually rolled leaves. They're modified leaves that used to be leaves but have been reduced because we want to conserve water loss. What are the structures that water is lost through? The stomata. If you roll your leaves and decrease the stomata, you're going to decrease the amount of water lost. A reduced number of stomata decreases the physical number of openings through which water can be lost and therefore will reduce the amount of water lost. Stomata are located in crypts or pits on the leaf surface, causing increased humidity at the opening. In a cross-section of these rolled leaves, you'll actually see that there is still an opening because we want gas to exchange because they still need to photosynthesize, but all of their stomata are in pits around the inside. Well, the inside of this remains relatively humid, and it therefore causes the water to not lose as fast or not be lost as fast, and they have these hair-like projections called leaf hairs that are going to trap and reabsorb the water that is lost, kind of like mini roots on the inside of these rolled leaves. A thickened waxy cuticle reduces water loss by increasing the barrier to prevent the water loss. Basically, high temperatures and high UV indices are going to not penetrate the leaves as well because you have an increased waxy cuticle around the outside. Hair-like projections on the inside of these rolled leaves are going to help trap the water vapor and reabsorb them into the plant as a way to prevent the water loss. Many desert plants shed their leaves or enter a state of dormancy during the driest months. Again, if we can shed the leaves and therefore shed the stomata, we reduce the amount of water lost and we can conserve as much water as possible. Cacti exist on water that the plant stores in fleshy, watery fruits or fleshy, watery tissues, especially in succulents, have a lot of thick leaves that are high in water concentration. They obtain this water during the rainy season and then store it year-round, or at least through the driest months. Okay? The last part is the CAM plants. This is the crassulation acid metabolism plants, and basically the only thing that you need to know from this particular topic is that CAM plants use an alternative photosynthetic process that allows for the reabsorption of water in order to basically absorb CO2 during the night and then store it to run during the day while the stomata can close. Normal plants or C3 plants are going to keep their stomata open all the time, which is going to cause an inflow of CO2 always to be present. That's also going to allow H2O to be released all the time. CAM plants, it's all about opening the stomata at night when the light intensity and the temperatures are lower. CO2 gets absorbed, it stores the CO2 at night, and then it uses the CO2 during the day when the stomata are closed. Okay? Really important adaptations for bryophytes and some of the CAM plants, okay, or the xenophiles. Those are all the stuff for this video. Bring your questions to class. See ya.